Well, okay. I like to start with karyotypes and chromosomes in genetics in general, although we'll be certainly talking about a lot of different things. Karyotypes are very visible and they're very not abstract, as opposed to a lot of other genetic things which kind of can be somewhat abstract. And it's good to think about the genome as a whole and to know what chromosomes are like before we get started with actual genetic crosses and things. So what we see here is a karyotype of a normal human female cell, and you can see some things about the genome just by looking at the picture. For example, the chromosomes are lined up and they're numbered, and each one is paired with one that looks very similar to itself. So we have 23 pairs of chromosomes. Because we're diploid, we have 2 of each chromosome. 2 of chromosome 1, 2 of chromosome 2, etc. The difference is the sex chromosomes, which is the 23rd pair. This is a female. She has 2 X chromosomes, which are located on the lower right part of the picture. So we can already see that in our normal female somatic cell, we have 46 total chromosomes, and they're arranged in pairs because we have 2 of each one. So one concept here is the concept of N. N is the number of chromosomes in the haploid chromosome set. So N for us is 23, and that's going to be an important number to remember from now on. Okay. All right, so in terms of how this is made and what we're looking at, we have to think about the fact that these chromosomes are very condensed. That's because the cells that these chromosomes came from were actually growing in culture in mitosis, so that they were captured at a stage just before they started to divide. So this is a point where the chromosomes are most condensed, they're visible, they're easy to see, which is in contrast to the rest of the cell cycle where they basically are dispersed throughout the nucleoplasm and you can't make them out at all. Okay, so I just said this, I didn't say homologs. We have the 2 members of the pair are considered homologous because they actually contain the same genes as each other, more or less, and one is from mom and one is from dad. Okay, so how these karyotypes are made is kind of amazing, really. We have to get a cell sample from the people, from this woman. We have to proliferate the cells in culture in such a way that we acquire a lot of cells that are actually dividing, actually in the mitotic part of the cell cycle. The cells are killed at that point, spread out on a slide. Notice the word spread here, in such a way that they're dispersed, and then they can be stained and imaged, and then somebody that knows what they're looking at can take these chromosomes, cut them out of the picture, and line them up to make the nice, neat karyotype that we saw before. So this whole idea of chromosome identification is important to the karyotype, because each chromosome, and its homolog, of course, contains different genes on it, and you want to be able to arrange them in pairs. Okay, so I just said this. Okay, so the staining method that's used is called the Giemsa stain. Well, it's one of many, but this is the characteristic one that's been around for a while that most people use. And it produces this characteristic banding pattern on each chromosomes, which clearly aids in an identification. And after you're identifying them, that's when you can cut them out and make the nice karyotype that we see. Okay, so how to tell which chromosome is which? Before these nice banding patterns were available, pretty much you could only go by the overall size of the chromosome and the position of the centromere along the length of the chromosome. All right, so what that means, of course, the centromere is visible as a constriction within the chromosome, and it occurs on a characteristic location on each of the chromosomes. So the centromere divides the chromosome into 2 arms, the short one being called P and the long one called Q. Some chromosomes that have the centromere smack in the middle, and those are called metacentric, to mean that it's in the middle. In those cases, then the P and Q arms are more or less defined arbitrarily according to the banding patterns. Okay, so the Giemsa bands that we see are characteristic, again, of each individual chromosome. And these ideograms are basically a representation of what those banding patterns are supposed to look like for each one. So your typical chromosome 1 has the banding pattern as shown by chromosome 1 here. And so when a cytogeneticist looks at the chromosomes and they're trying to identify them, they would look for the banding pattern that's characteristic of that particular chromosome. Within these ideograms, the centromeres are lined up on a horizontal line. Notice also the X and Y chromosome on the lower right. They're very definitely distinguishable from one another, but they're considered as members of a pair because those are the sex chromosomes. Okay, here's two other karyotypes, just to have a male karyotype here. The karyotype from the normal female is shown at the left. You can see the two X chromosomes on the lower right, and you can see that she has 23 pairs. And the male also has 23 pairs, and his sex chromosomes again are shown, the X and the Y on the lower right. Sometimes those are shown together as a pair, and sometimes those are shown separately as shown here. Okay, so ideas so far. The chromosome number N is the haploid number. N is 23 for human. Ploidy, we are diploid, meaning we have two of each for 46 total chromosomes. We could identify the chromosomes based on their size and their G-banding patterns, and of course the position of the centromere. And the idea of the word ideogram is a representation or a drawing of what somebody is supposed to see when they look at these chromosomes. Okay, so now for a few questions about this to see if you can expand beyond human to other organisms. Here's a karyotype of a dog. Dogs are like people in having an X and a Y chromosome to determine their sex, but they obviously have a different number of chromosomes from what people have. These are arranged again in pairs as if it were a human one, but it's a different number of chromosomes, quite definitely. So if N is 23 for human, N for dog is 19, 20, 39, 40, or 80. Okay, it's 39. An organism is diploid and has a haploid chromosome number N of 5. The number of chromosomes visible in a karyotype of this organism is 5, 10, 15, or 20. Okay, the answer is 10, right? Good. Okay, here's a ploidy question. This karyotype is from a triploid cell. Could it be a human cell? Yes or no? Okay, the answer is yes, because there actually are 23 different chromosomes here arranged in threes for a triploid. So this would be a human cell that is actually got 3 copies of each chromosome instead of the characteristic 2. So now I'd like to move on a little bit to talk about how much DNA is present in the karyotype, mostly to remind ourselves what we're looking at in a karyotype and to understand more about the cell cycle. Okay, so here's your typical cell cycle diagram, and it's divided into phases. The G1 phase is the cells that have basically just divided, and then they're going to grow for a while to be able to divide. So then after they grow for a while, they will enter the S phase, and that's when DNA replication will occur. Following the DNA replication phase, there's another gap, or G2 phase, where cells grow further and prepare for mitosis. And finally, the cells will then condense in order to undergo the mitotic divisions to produce 2 new cells and then start the cell cycle all over again. So remember that when we're looking at karyotypes, we're looking at cells that are actually in the mitotic phase of a cell cycle. And that's important in terms of thinking of how much DNA and how many chromosomes, etc., are present in those cells. Okay, in order to understand what we're seeing in the karyotype and understanding what's going on in the cell cycle, we need another word, and that's called C-value, which is related to N, but not at all the same thing. Okay, C is the amount of DNA in base pairs in one haploid genome. And for us, that number is 3 times 10 to the 9th base pairs. Okay, it's important to recognize that this is the amount of DNA in one complete haploid genome, not for each chromosome, right? So the chromosomes, there are 23 of them, different ones. And so that 3 times 10 to the 9th base pairs is divided up among or distributed among those 23 chromosomes. So it's basically the entirety of a haploid genome, but that's of course not what's present in our cells because we're diploid, and we also have DNA replication to consider. Okay, each chromosome at this point is made up of one chromatid, which is DNA in complex with the histone proteins, etc. Okay, so at the beginning of the cell cycle in G1, after mitosis is finished, of course we're diploid, so we have two N chromosomes, 46 total in each cell, which is to say N is 23 and we have two of each. Okay, in terms of DNA, relative to the haploid DNA content, C is one haploid equivalent of DNA, but we're diploid, so we have twice that much of DNA in our cells at the beginning of the mitotic cell cycle. So that's 6 times 10 to the ninth in each cell. Right, DNA replication has not yet occurred, so each chromosome contains one long double-stranded DNA molecule that's one chromatid. So there's 46 chromatids in the cell, 46 chromosomes in the cell. Okay, so then we're going to go into the S phase, and DNA replication will be happening. Remember semi-conservative DNA replication. Each strand is a template to create a complementary strand. Replication begins at origins that are spread out along the eukaryotic chromosome, and it continues until each chromosome is made, each molecule is made a replica of itself, essentially. So that's what happens during S phase. Of course, you have to do that to prepare the cell for the actual mitotic division, which is what's going to happen next. Okay, so as we're envisioning what's happening here, at the beginning of S phase, each chromosome consists of one chromatid, as depicted. And then during the S phase, the strands are duplicated to form a chromosome which has two chromatids on it, right, each attached to the same centromere. Those are sister chromatids because they arose by DNA replication, semi-conservative replication of each other. It still counts as one chromosome, though, because there's only one active centromere in the cell. But we do have two chromatids. Okay, so after S phase and during mitosis, again, we've got to do our arithmetic. We're diploid, so we have two N chromosomes. 23 times 2 is 46 total. Before DNA replication, we had 2C of DNA because we're diploid. After S phase, we now have 4C of DNA because the DNA replicated. Okay, counting chromatids, and this gets a little confusing. Each chromosome now has two chromatids. There's 46 total chromosomes. So we actually have 92 chromatids in each cell. But we still have 46 chromosomes, but each of them has two chromatids on it. So each chromosome, even though it's got twice as much DNA on it, twice as many base pairs, it still counts as two N chromosomes because the centromeres haven't divided yet, and even though we have 4 N chromatids. So that can be a little bit confusing. Now, our cell that we've killed to make our karyotype didn't get to do this, but we started our karyotype at this stage in the cell cycle when the cells were at mitosis, the two chromatids were still attached to the same centromere, and so we still had 46 chromosomes in our cell, even though we have 92 chromatids. What would have happened had we not killed the cells, but we did, is they would have separated. And so at that point in anaphase, each chromosome becomes separated. Each chromatid separates from its sister chromatid, and that gives us actually 92 chromatids, but also 92 active chromosomes in the same cell. So we have to, when we're doing our karyotype arithmetic, consider what stage in the cell cycle we're at. And it's basically after replication, but before the sister chromatids have separated. This can be confusing. All right, partly, I think what's confusing about it, all right, so after replication, the cell has 4C of DNA, 4 N chromatids, 2 N chromosomes. But then the cell will divide, not in our karyotype cell, but the cell will eventually divide to restore the diploid chromosome number after that fact. Okay, the reason this is confusing is you can't really see the sister chromatids in the pictures. So people do confuse the homologous chromosomes from the sister chromatids sometimes, and that can be confusing. All right, so here we have the karyotype we looked at before. It's got its 23 pairs all lined up nicely. What you cannot see here, but is important to recognize, is that each one of these chromosomes has actually got two sister chromatids on it, but they're squashed together to the extent that you cannot distinguish that they're there. So we actually have 4C of DNA and 4 N chromatids here because of that fact. Okay, so here is a picture just to show you what it really looks like, and this karyotype was done in a different way, in such a way that the sister chromatids are visible. So each pair, you can see each chromosome, the two sister chromatids are starting to separate out a little bit from each other. So this picture actually enables you to see all the sister chromatids, which were not visible in the previous picture because they were still so squashed together you couldn't distinguish them. But it's important to recognize that they are there, but they just haven't separated yet. Okay, let's have a few questions about this now. The C value is the amount of DNA present in one haploid set of chromosomes. For us, C is 3 times 10 to the 9th base pairs. Therefore, the amount of DNA present in our somatic cell karyotype is 3 times 10 to the 9th, 6 times 10 to the 9th, 12 times 10 to the 9th, or 24 times 10 to the 9th. Okay, so the answer is that it's 12 times 10 to the 9th. If C is the amount of DNA in one copy of the genome, each karyotype contains 2 of each chromosome, and each chromosome consists of 2 side-by-side sister chromatids. So the amount of DNA in the karyotype is 4C. So for us, the total in the karyotype is 12 times 10 to the 9th base pairs in the picture. Okay, here's another dog question. A karyotype of the dog is shown below. Sex determination in dogs is like sex determination in human. Is this dog male or female? Well, the dog is male. It has an X and a Y. How many chromatids are present in the dog karyotype shown? Okay, we can know that there are 39 pairs of chromosomes. Each chromosome is got a homolog, and each homolog has a sister chromatid. So the answer is D, 156. How many chromosomes are present in the dog karyotype? The haploid number is 39. We know that it's diploid, and it's 78. So there's 78 chromosomes present. Even though each chromosome is actually double, duplicate, it's still only 78 real chromosomes. Okay, so we're going to do a worksheet about bananas, and I think you know everything you need to know about that. So I'm going to post it, and you can download it, and we will go over it next time, and we'll turn it in for 1 bonus point. Yay!